See if this works.
See if it works.
See if this works.
See if it works.
Yeah, looks like it's working now. Okay, it looks like we've got a small but strong contingent right now. Might as well get started. Okay, so thanks to everyone for being here for our first talk of 2022. I'm very happy to have Milo Lin here to give this talk. So just a word for any newcomers about what we're trying to do. So Milo, I assume it's okay if people interrupt you during your talk with any questions. To do that, you can unmute yourself. I'm gonna mute somebody. Okay, so yeah, maybe everyone keep yourselves muted until you've got a question. So you can then, if you have a question, just unmute yourself or use the hand raise feature, and then one of us can call on you. So the format of these things, what we like to do is have the talk part, of course, but then towards the end, maybe reserving the last 20 to 30 minutes for a broader discussion. And we'll switch off the YouTube streaming and recording for that part. So maybe we can talk a little bit freely about our opinions about science. So, okay, and a lot more people rolling in now. So Milo, I'll just go ahead and introduce you. So Milo completed his PhD in biophysics and physics Caltech in 2012, where he was the winner of the Clauser Prize for the most significant PhD thesis at Caltech. From there, he went to Berkeley, where he was a Miller Fellow with David Chandler. And then from there, in 2016 or 2015, he went to UT Southwestern Medical Center, where he's been assistant professor in the Green Comprehensive Center for Molecular Computational and Systems Biology and the Department of Biophysics. So very excited to have you here, Milo. Take it away.
Thank you, guys. Thank you, Karna. Thank you, Simon, for the invite. And my understanding here is that also for the talk part, it can also be not just about one kind of scientific topic, but also like weaving in maybe some themes of like what my lab is about and the way we kind of think, and some kind of broad guiding goals or principles that we operate or like to operate under and are striving for. And I guess my kickoff slide is this classic paper by P.W. Anderson that has been very influential in defining the birth of the field of systems biology. Now, I'm in the Green Center for Systems Biology here at UT Southwestern, and I think the one very powerful motivation for systems biology is kind of encapsulated in this idea, which I've highlighted here, but that boiled down to really says, if we're going to understand complex systems, the way to do that, or the pathway to doing that, is to maybe find a different set of collective variables, different set of variables that you can examine it in. And from that different set of variables, from that different perspective, maybe the relationships will be a lot more clear. And the question in the quest is always, is there such a set of, is it just intrinsically hard to understand and complex and just irreducible? Or are there ways of looking at a problem in a different way, mapped to a different set of variables, coordinates, that allow for systematic simplification, at least for the class of problems that we're interested in here. We're interested in the biological complexity, right? Complexity, particularly at the molecular and the cellular scales. So that is kind of a theme of what my lab tries to do. And maybe I'll lay it down kind of, or lay out a course of action that we like to pursue for various problems that we work on, which is basically these three steps, right? So first, through some way or another, find some set of effective, usually collective variables for a system that is a different way of looking at the system. And hopefully that allows for not just a different perspective, but a much simpler perspective on the system. Maybe things that are complicated in the naive variables are not as complicated in these effective variables. After doing that, what we'd like to do is to predict something that's not obvious without this mapping to these new variables, right? So if we're just getting these collective variables and showing some fancy math, that's not enough. We want to predict something that's just hard to like intuit or hard to kind of even see without seeing in this new way. And then I think the even more challenging thing, but I think the thing that ultimately will be important for having a connection and an impact is that if it's really a good way of looking at something, you should also, in principle, be able to use this new framework, these new variables, to build something that is also hard to build. If you're only thinking in the old or the direct degrees of freedom, right? And the biological problem that I'm going to focus on for this talk, we've done the first two, and we haven't yet done the third thing yet. But to motivate that this is in principle possible, I'm just going to briefly give you an example of another problem that we have been working on in the lab, in which we believe we've hit all three of these things, and to show what that could look like, just to motivate what we're aiming for the meat of the talk that I'll be spending most of my time on. Okay, so a little bit of a aside as an illustration of what this might look like. So for this problem, we're trying to understand information processing in a neural network, okay? And what we've done very briefly was to find the collective variable, not in terms of the weights of the synapses of the neurons, but in terms of the decision boundaries the neurons make, and then how those decision boundaries can feed downstream to form hierarchically convex shapes in input space, so convex regions in input space. So here the collective variables are decision boundaries, and the new effective variables are decision boundaries, and the collective variables are these convex shapes that can be constructed from decision boundaries. So doing that, we can first predict certain things that we would like to know. So here are just four graphs illustrating four analytic results about the what we call the comprehension capacity, but it's just another way of saying how many different decisions you can make about a problem given certain resources, right? So if you are given a certain number of neurons in a neural network, and I were to give you, let's say I would ask you, let's say there's this neural network has to predict the output that correspond to some complicated function that can weave in and out and be positive and negative and intersect the zero line at however many places. We ask the question, what was the maximum complexity as measured by how many zeros this function has, that this neural network could possibly compute, right? This is what we call the comprehension capacity. It's a measure of how many basically decision boundaries there are in one dimension. And what the result basically is to say, okay, let's consider the two things we can control in terms of the resource of a network. One is how many neurons there are in the neural network, and that's shown in the x-axis of these plots, how many neurons there are. And then the four different, sorry, that's shown in the there's two different, let me back up. There's two different ways that neural networks could be different. One is that the neurons themselves could either be digital or analog. Digital means that they only output 0 or 1, and analog means that they can output some real number as their output. Another feature that a neural network can have is that they can either be layered like this, that each layer only feeds information to the next layer and not to any other layers, or they can be completely connected. These are two qualitative classes of neural networks. So that breaks down into 4 categories. And this, I guess the simplest are neural networks in which the neurons are all digital, and the neurons are only put in layers, okay? And what we basically show analytically through the new collective variables of these decision boundaries is that if you don't have either one of these 2 properties, so if you're digital and if you're layered, the number of decision boundaries grows linearly with the number of neurons. But if you can violate any one of these 2 constraints, so you can be still layered, but now your neurons can be analog, or your neurons are still forced to be digital, but now you can have a completely connected network, then the number of the comprehension capacity as measured by the number of decisions boundaries, it grows exponentially with the number of neurons. So that is the prediction, and here is just showing, like in red is what happens if you just randomly simulate huge ensembles of neurons, and then the blue and the green indicate what happens if you now do a computational evolutionary simulation where you have populations where you evolve to have the maximum number of decision boundaries. And you can see that, like as the number of neurons gets larger, these evolutionary simulations can start to can still hit, but then start not being able to hit the analytic bounds. But this is a little bit older graph. We actually have it now updated so that it hits the bound higher up, but it gets harder and harder to evolve systems that are at the maximum bound, right? But I think more exciting is the third step, which is not to now use these collective variables to, oh, go ahead. Someone had a question.
Yeah, I'm just wondering, how do you get the analytic bound? Is that like, you can just write down equations and solve them?
Yeah. And I because this is not the main, I would be happy to talk about this other project offline, but because this is not like kind of the main topic, basically, yeah, it's you just write down the equations and you ask like, and you prove that this is the maximum number of boundaries they can achieve.
And does that like analysis, can that help you build a network or not?
Yes, exactly. So this gets me to my third part, which is now, can we build something? And so using these collective variables, we've figured out a way to build an artificial neural network that is built using a hierarchical framework, using these decision boundaries, and now is not built on gradient optimization. So they're not trained via backpropagation, but yet on a set of benchmarks, machine learning benchmarks, they perform just as well as like backpropagation, like deep neural networks. But they give you very different insight because they're interpretable neural networks. So unlike a deep neural network where you don't know what each neuron is doing because it's a black box, because each neuron is participating in some high dimensional gradient, here you can assign meaning to both you can assign meaning to the neurons and the different layers. So the first here are the differential neurons, and you can tell that each differential neuron is actually distinguishing between 2 different class, like a one type of 9 and another type of 5, right? But I think more interestingly, you exhibit that in the deeper layer, you now have this modular structure where things that are responsible for the same kind of output tend to connect together. And that's a feature that's not found in existing deep neural networks, where everything's kind of like connected to everything else. And it's also reflected in the firing patterns of these neural networks. So we are now using this as a much closer model system to actually model information processing in biological networks, because these networks have the property that they exhibit these properties like modular connectivity and both distributed and localist firing patterns that are missing from deep neural networks. But the other thing that we're able to do is now that these neural networks, which we call essence neural networks, are also able to generalize out of distribution, which is something that existing deep neural networks can't really do. And they can therefore solve like symbolic problems, and they can also solve complicated problems by only training on very simple problems, much like you would train a human, right? You would train a human on very, very simple illustrative problems, and then once they get the concept, they will abstract it and be able to solve things out of distribution. And this plot is just showing that on two problems. One is an NP complete problem, and the other is a vision problem, in which is supposed to say what is horizontal and what is vertical, right? It's only trained on these purely horizontal and purely vertical lines, but now it's being tested on these things that are supposed to ask, has it grasped the more general concept of the aspect ratio? So boxes and zigzags and diagonal lines, etc., and images with noise in the background too. And what's interesting is that this ENN actually generalizes to all these cases perfectly, has zero error, whereas as a control, the gradient networks generalize very poorly once you give it things it hasn't seen. It also does not solve these symbolic optimization problems. But also these things ENNs are so interpretable that we've written a automatic, what we call a condenser, that takes this neural network, like just scans it and automatically produces Python code that's equivalent to the neural network. So that's how interpretable they are. So this is actually the code. So this has 1.4 million parameters, but the equivalent Python code is 54 lines, and it's automatically generated by our condenser. So now you have a completely end-to-end from the pure data to the generalized abstraction of the pattern in the data without any human intervention, right? So this is we're trying to build towards like an automatic experimentalist. But I think this is just like an illustration that if you then use these collective variables, you can not only predict new things, but you should be able to then use them to build something that's hard to build, without that framework. So that takes me to kind of what we like to also do, which is the topic of this talk. And I think,
again?
Go ahead.
Yes. What are the collective variables in a neural network?
Oh, so here the collective variables we're arguing is this hierarchical framework of decision boundaries, right? So these are the effective variables that you consider. These are decision boundaries, but decision boundaries on the first layer become convex shapes in input space in higher layers. So these decision boundaries bound convex shapes, and then you can put many convex shapes together to make arbitrary, very complicated non-convex shapes that are supposed to be your supervised, say, learning task, right? So the collective variables are decision boundaries, and they're and the generalization of many decision boundaries coming together, which are these convex shapes.
I see.
In input space.
Cool, thank you.
Okay. All right. So here, I think for more of a physics audience, I think I thought I would talk about something that's more a work in progress in terms of we're not quite there at the third part of building something, but I like to just show our results on thinking about this problem and the predictions that we're able to make, right? And here it's a little bit just to copy what the title of Anderson's paper, and here we're thinking about not just that, basically that having more things may necessitate having new collective variables that end up bringing to bear qualitative different behaviors than having few things. Here, the theme of this is that using the collective variable, which I will introduce in the next couple of slides, we can say the same thing about energy, that a system that's driven close to equilibrium is fundamentally qualitatively different from a system that's driven far from equilibrium. And we can see how that is so using the collective variable, which is the circuit mapping that I'll show. Okay. All right. So, I mean, nobody here needs too long of an introduction of the the challenge and but the opportunity of complexity that is biology, right? And one way that we like to think of biology is that it is, and the way that's non-trivial and interesting is that it's energy plus complexity, and all these things. I'm illustrating various classes of behavior in the cell, from signaling to building infrastructure to transport to replication. These are all driven far from equilibrium by constant consumption of energy. The quest for non-equilibrium statistical physics is to really be able to understand these systems, to extract unifying principles, and to be able to predict them in non-trivial contexts, right? So, but what makes them difficult is that because they're far from equilibrium, there's relatively little mathematical machinery that we can use to simplify the behavior of these systems and to predict how they would differ if you change something, right? So here I'm just now motivating thinking about these non-equilibrium systems in terms of their state space. So each node here is a possible state of your complicated system, and you can hop from one state to another, and you can reversibly hop back. And in the absence of driving, right, the system would reach equilibrium, and it would reach detail balance, so that the net forward is always equal to net backwards. But because of sources of energy, which I represent as arrows here are sources of energy that come in and break detail balance locally, and therefore leads to breaking of detail balance globally. This would correspond to, like, say, binding ATP right here, right? ATP that normally would be very, very low concentration because it's a high energy state, but it's pumped in to maintain a high concentration of ATP that can be used to drive these systems, right? And these circles denote mass sources and things that you can have states that come in or disappear as well. You can have the creation and destruction of matter. And because these systems are now driven far from equilibrium, we need some way to simplify them. We need some good collective variables. And what we argue here in this paper is that the circuit mapping is a good collective variable. So a set of collective variables. So this is just a kind of intro of how we do this mapping. So we first in the state space, we take the passive elements. For example, here's the state M. We can define what we call a probability voltage. It's just a scalar quantity that is a state variable of the state M. Okay, and here it's equal to its probability times e to the beta gm. Gm is the free energy of the state M in the absence of driving. So you have a system and you don't drive anything. You let the system reach equilibrium. Now you can define the free energies of all the states, right? These states, they can be microstates, they can be macrostates, they can be multiple little states lumped together. You can define the free energies of all the states at equilibrium before you drive the system at all. And then you drive the system, and now this state will have probability P, but the GM is still the free energy of the bare undriven reference system. Is that clear? The definition of the G? The G is always in reference to the reference undriven equilibrium system. But the P is a probability of interest, which is the probability of state M when it's driven, okay? So the state variable, the voltage, is this. And you can see that at equilibrium, this is PM is proportional e to the negative beta GM. And this thing is just becomes like 1 over the partition function for every state. So what you can see is that at equilibrium, it just says that each node has equal potential, okay? But far from equilibrium, PM is not equal to e to the negative beta GM. And that's when you have different potentials, probability potentials that will drive, these potentials then drive net fluxes of current, probability current between states, okay? Okay, so the other passive element is again defined in terms of purely equilibrium parameters. So if you have a resistor between state M and N, and that's defined to be e to the beta GM over the equilibrium reference rate constant, okay, of going from state M to N, KMN. And it is equal so Rmn is equal to Rnm. So this resistance is symmetric. So it doesn't matter if you define it from M to N to N to M, you have the same resistance. That's going to be important to have a lot of the properties that I'm going to talk about later. We would have to have symmetric resistors, okay? Just like the resistors are symmetric in normal electronic circuits, okay? All right, and then the driven elements, there's another mapping to mass sources, which I'm not going to talk about. Let's just focus on the energy source, where what you have is that you have now effectively a battery from state M to N, because you're directly driving that transition. And you're driving that transition with an excess rate constant alpha, okay? So if alpha is 0, you're not driving this transition. When alpha is not 0, you're driving this transition. And so this voltage drop of this battery is equal to alpha over K, which is the driven divided by its original equilibrium rate constant times the voltage of the driven state. Okay, so it's it has, aside from this aspect of negative feedback, it's just like a DC current, just like a DC voltage source. Okay, it's like a battery. Okay. And you can also see that, like the in terms of these kinetic parameters, alpha and K, you can define what the chemical potential differences driving the system by it's equal to log of 1 plus alpha over K. So again, when you're not driving it, there's 0 chemical potential difference driving the system. Okay, and these delta mu's, these chemical potential differences, are what drives the system. Okay, so this is the mapping. Okay. Why do we have this map? Why do we choose this mapping? Because if you choose this mapping, if you choose this set of effective parameters, effective variables, then this equation is true. And this equation is basically the voltage equation that obeys Ohm's law. Okay, so that is that the voltage difference between any two states, J and I, right? Remember, this Pj times e to the beta Gj is Vj, the voltage, the probability voltage of state J. So this is just saying that what the voltage difference between two states J and I is equal to the sum along any path from I to J of this whole quantity of, like, all the batteries in that path minus the resistors times the currents in that path. So exactly, this is just exactly Ohm's law, right? And it's true along any path. And this is also true, it doesn't have to be at steady state. So this obviously applies at a non-equilibrium steady state, but it also applies when you haven't even reached steady state. Just like the voltage equation applies when you haven't reached steady state, okay? And so this is why we make this mapping. We make this mapping because Ohm's law is true. And because Ohm's law is true, and this we have this mathematical isomorphism, we can now port over a lot of the simplifying power that we have from electronic circuits, right? The easiest being that you can now sum, if you have a whole bunch of passive dynamics, so you're driving the system by ATP binding here, and there's all sorts of stuff that happens. Your molecule could be binding other things, undergoing conformation, a whole nest of transitions that are not directly driven. Those transitions corresponds to just resistors, and you can combine them in series and in parallel, right, to make them into an effective Thevenin equivalent resistance, right? That is, but you can also port over lots of very powerful circuit theorems that were derived for circuits that I'll talk about a little bit later. So now you can see how this mapping could allow for some simplification and to getting out some analytic results for biological systems. Right. So basically, if you look at the cell as a whole bunch of comprised of a whole bunch of these underlying chemical engines, right, that do bind something, maybe make some chemical change, release something, and then is ready to go again, right? There's this, I call all of this signaling, right? It's also connected to these processes of metabolism that take external energy and then converts it to energy currency to be used throughout the cell. And then of course there's assembly and transport. The assembly process itself is directly driven, but so is the transport of stuff along these cytoskeletal elements, right? Now, if you now just zoom in at one of these things, let's say this is a signaling molecule, a signaling process, this is what it actually looks like, right? So you have the thing it binds to its ligand, right? And but now it binds to ATP, and that's the driven transition. Now it can hydrolyze ATP, and there is a chance that after it hydrolyzes, it just falls off and the whole reaction fails. It's a futile cycle, or it can succeed and actually use this energy to catalyze or convert this reactant into a let's say here in signaling, it's an inactive form to a square to an active form, a triangle, and then it can release it, etc. Right? So, but that just looks like this, right? And then you can see that you can combine all these resistors together to one effective load resistance, and you know exactly what this is in terms of the microscopics. But if you didn't know what the microscopics are, you can also measure this. It's just the linear response. If you have some complicated process and you just perturbatively drive it, and you look at its current response, the ratio of how much you drive it to its current response, this linear response coefficient is also the total resistance. So you can get it from analytically from knowing the microscopic variables, but if you don't know what the microscopic variables are and you want a number, you can also measure it from a perturbative measurement so this RL can be gotten from a linear response measurement, but the theory applies far from equilibrium, right? So this is the same resistor parameter that's used. So the previous Ohm's law thing is not a near equilibrium result, but it's true for arbitrary levels of driving. Okay, so oops, I think I went low. Now I can't go back. Okay, this is somehow I've gone into this is a highly irreversible presentation. Let's see. I don't think I'm okay, maybe I'll figure out in a bit, but you guys briefly saw what the diagram was for microtubule self-assembly, right? But this is what it looks like when you map it to a circuit. And then you can solve this analytically. So it's interesting that it just breaks down into a superposition of two exponential decays. One is the pure equilibrium exponential decay that depends on the equilibrium free energy of binding of each monomer. But then there's this non-equilibrium exponential decay that appears here. Notice when alpha is 0, this thing just goes away. You just have the normal boring thing, right? But now when alpha is not 0, when you're driving this system, each one of these, this is a driven process, this piece plays a part. And this D is a function of the variables, but you notice that it's also a function of how far you're driven from equilibrium. But the upshot of being able to get an analytic expression for this is that we can now understand that there is a interesting transition from the near equilibrium regime to the far from equilibrium regime. And I think this is biologically of interest because it actually encapsulates a biological capability. What is that capability? We all know that at equilibrium, a catalyst cannot change the steady state outcome. The catalyst only makes the reaction go faster, right? So it just lowers the energy of the transition state, but it doesn't change how much product you make. That is what a catalyst does. That's a equilibrium. And that's borne out here by this blue line, right? So when alpha is 0, you have this completely expected equilibrium behavior that, oh, sorry, I should define what FCAT is. FCAT is the frequency of catastrophe. So these microtubules, they grow on one end, they grow like this. And then when there's a catastrophe, a banana peels. So the way that it grows is generally different in path from the way that it falls apart. And this is, I mean, this is a very striking violation of detail balance, right? So F is actually the frequency that you can get this thing to catastrophe. Okay, and that is something that you can control just by if you just dope in some molecule that messes with the growing cap of the microtubule, you can control this rate constant of catastrophe. right? But notice you can put in something at very, very low concentrations because it's acting like a catalyst, and it only acts at the tip of the growing microtubule. It acts and is not used up. It can act again, right? So it has a power of a catalyst. You can have a lot of effect at very low concentration because it doesn't have to act stoichiometrically with what it's targeting. That's the whole power of the catalyst. But at equilibrium, that power is limited by the fact that you can only make things faster. You can't change any outcomes. But now, when you're driven, when the system is driven, suddenly your catalyst can not only changes the rate of the reaction, it actually changes the steady state outcome. So what I'm plotting here is the from this is the probability of having a microtubule length L. Okay, so from that you can calculate the mean length of the microtubules, and you can see that as you start driving a little bit from equilibrium, you start violating this. You can have a very small effect by changing this catalytic rate constant. Okay, very small though, because you're still near equilibrium, and it turns out that this effect is logarithmic. However, at a transition condition that's set by the parameters, you transition from a logarithmic dependence on FCAT to a linear dependence on FCAT. Now, changing this catalytic rate constant linearly tunes your mean microtubule length at steady state. So you're transitioning from this category of regulatory power near equilibrium, which is essentially non-existent, to this category of regulatory power at far from equilibrium, which is you can linearly change a steady state outcome. Here, it's actually important that very quickly, the microtubules very quickly go from one change in length by a factor of 2 to 3 from mitosis to interphase, okay? And that happens largely just through tuning this FCAT. And it can happen very quickly because it's just happening at the tips and it's a catalytic event. But you now have this capability open to you far from equilibrium, and there is a relatively sharp transition from one to the other, so that if you didn't know any better and your system had evolved to just be here, you would think, catalysts can't change the outcome. They can only make rates faster, right? But if you just evolve your GTP concentration inside the cell to be a little bit higher, you suddenly gain the power that your catalyst can actually tune your outcome. Okay, so this is what this gap is one illustration of what we mean when we say more energy is different. That being driven a little bit further from equilibrium, you access a qualitatively different regime of behavior and tunability. Okay, the second example. How am I doing on time here? Sorry, I don't want to. I want to give some space for the. Okay, I think I'm this 1:07. Okay, yeah, I think I'm doing okay on time actually.
Yeah, maybe about 25 minutes.
Okay, perfect. So the second example is kinetic proofreading. Here you have the ribosome, and what you ideally want is you want to have like you can have the you will bind to another tRNA complex, right? That has the right codon pairing with the next codon in the RNA, which is represented by this black fork trident here. You guys can see all this time you guys have been able to see my cursor, right? Okay, thank God. All right, so this black trident here, right? That is a codon match for this, so it binds well. And because it binds well, DNA complements, is all good. Then it will hydrolyze the GTP. And now when it hydrolyzes GTP, there's a chance that this covalent bond doesn't get made and the thing falls off and it's just a futile cycle. So you've just wasted that energy. Or some of the time you actually get a successful thing and you have a just elongation of your protein. You've made another amino acid in your protein correctly, right? On the other hand, you might have an imperfect, might have imperfect codon match denoted by the red trident. So this has less energetic favorability than the perfect match over here, but still enough binding that it might stay for a little while. So you can still bind, and there's still some chance it'll hydrolyze the GTP, and hopefully it'll just fall off so you don't make that error. Oh, sorry, the fall off is this side, my bad. So hopefully it'll just fall off so you don't make that error, but there is a chance that it won't fall off and it will link the incorrect, this triangle amino acid, which is the incorrect. So you made an error in translation, okay? So you want to maximize like this, and you want to minimize this. So this is what it looks like as a circuit. And I'll just rehash some very old results already established by Hotfield in the 70s on kinetic proofreading, that if the energy of binding between the correct codon and the incorrect codon is delta, okay? From equilibrium arguments, you would say that you would make an error proportional to e to the negative delta over kt, because that's the energy difference between the correct and the incorrect. So that's how often you will like sometimes get the incorrect. But Hotfield showed that through this active process of consuming energy and having things fall off, and having things have the opportunity to fall off in multiple stages, you now can have this maximum outcome where it's actually 2 delta. And that makes a difference between something that's like okay, but like into something that is what the level of rigor that's needed in our cells, right? So, okay, this has been established, but what we're able to show now using this mapping is that there is something missing here, okay? So it's, yes, it can go to e to the at the maximum possible, but it actually has to sacrifice something to achieve this, this maximum bound. And what it ends up sacrificing is what we call omega, which is the waste, okay? So the waste is another metric that one could use to evaluate how well these things do. So the error is clear. It's like here the error is like I2 over I4, because I2 is the correct current of I2 is the current of the probability current of making the right thing, and I4 is the probability current of making the wrong thing, okay? But now you have to consider the waste is that you can have low error but have high waste, in that you have lots of I1 and I3 where things are just falling off all the time and you're just wasting the energy. So there is now, to make the picture complete, it's not just a constant, but you're only able to get at the maximum, the minimum error when you're at essentially the maximum waste, okay? So the prediction here is that basically the log of the error plus the log of the waste is constrained now by this 2 delta over kt. So there isn't this error waste trade-off, and here is just shown from experimental data. So the lines are this equation, and then the points are the measured the error and waste as obtained from this paper. So biology does seem to be at the limit of what it can do in terms of this trade-off. It has optimized the parameters in the system, okay, such that it actually hits this optimum trade-off. But again, here it's another example of more energy is different. We've now quantified how error and waste gets traded off, but the fact remains that you go from a near equilibrium minus delta, e to the minus delta over kt error, to a far from equilibrium minus 2 delta over kt. And we can also, in this theory, show how this transition occurs, how you go from near to far from equilibrium. So these are 2 examples of more energies different enabled by this framework. And but we know that in this framework, we can get analytic results in the sense that if we constrain the topology to be this, right? It's analytic in the sense that the results are true for all values of the parameters. But we would like something even more general than that. We would like something that's true for like any possible system, not just kinetic proofreading or assembly, any possible topology of state space, right? So how general is this principle of more energy is different? And that will be the last piece of my talk. So I think we're still doing pretty good on time. All right, so okay, so to kind of motivate that, I think the first example of we found this is really the birth of thermodynamics, right? For a long time, we were just trying to design heat engines that got better and better, and they each had like very special names and very complicated designs, and they did get better and better, right? But then, Carnot showed that basically the best you can do in terms of the efficiency is set by T0, which is the ambient temperature. That's the exhaust temperature that the exhaust heat is coming out, and then delta T is the temperature difference between the exhaust temperature and the temperature in which the high temperature like furnace or whatever that you're getting the energy from, right? So this is the relation, and that sets the best you can do. So if you're getting close to this already, you should stop tinkering with the design of your machine and start trying to like get things that can hold really hot things, so you can up the delta T. All right. So but what's interesting here is that in this case, right, it's what we call a Michaelian equation. That T just goes like this. So there's no special transition delta T. Like the higher the delta T, the more efficient it is. But you actually the slope is the highest at low delta T, and the slope, it just goes like that, right? As the efficiency tends to 1. So it's kind of a boring, what we call Michaelian function. Nothing interesting. There's no transition. But going back to the biology does not run on heat engines, it runs on chemical engines, right? So can we also think about a way to set an upper bound on the efficiency of chemical engines, also regardless of the mechanism? So let's imagine what a chemical engine could look like. So you have now not a delta T driven thing, but a delta mu driven thing that we call the battery. And the battery has its intrinsic resistance, just like the battery in your flashlight has its own intrinsic resistance. You would like all of that energy to be burnt in the resistor of the light bulb, but actually some of it, just by engineering design, has to still be dissipated in the battery, right? So the battery has its own resistance, just as parameterized by the rate constants, as I showed in terms of how the resistors are defined. And now it's driving some load process that can be composed of some arbitrarily complicated, state space where the machine can like do all sorts of things, and right, and doing this walk, coming back, it's performing some useful function. It could be a motor, it could be to assemble something, or it could just act to maintain some steady state pattern that would be impossible at equilibrium, right? What Prigogine called these dissipative structures. Okay, so here's a schematic of a chemical engine. And I'm not going to go through, in the interest of time, kind of the derivations of this, but using a very powerful theorem called Teligens's theorem, and making use of the circuit mapping. So you map it to the circuit, you use Teligens's theorem, and then you take advantage of other circuit properties. You can derive this equation, which is the kind of the parallel of Carnot's relation, but now for chemical engines, okay? So now the maximum efficiency of any chemical engine, right, is less than, okay, you see that just like in the heat engine, the delta T's appear there. Here is depending on how far from equilibrium you are, the delta mu's appear. But now it's also dependent on some coarse grain aspects of the battery resistance and the total load resistance, right? The Thevenin equivalent resistance of your entire load, this RL. So there's some actually, it's dependent on the circuit properties in that way, but this applies to any chemical engine with any circuit, right? That's okay, but what this looks like is that this is actually a sigmoid function. So unlike Carnot's equation, which just is this boring Michaelian thing, this thing actually has an inflection point. So if you plot this, so here what I'm showing is the x-axis is delta mu, how far away you are from equilibrium, right? And then the y-axis is the chemical efficiency, the max chemical efficiency, and the different lines from gray to orange just denote different ratios of your load resistance to the battery resistance. This should be RS. Okay, sorry for the confusion. This should be RS, the load of the source or the battery. This is RS over RL. Okay. And you can see that you can see where this inflection point happens in these curves. And so the inflection point happens from transition from when your whatever process is just like not efficient to when it can be efficient. Okay, so like for example, for this one, that so these white lines actually corresponds to the measured values for this diene, which is the only one that you can track multiple points, because you can then change the ATP concentration and get a measure of its efficiency, walking efficiency, as a function of ATP concentration. Okay, but we can see is that if you're at this gray line, for example, right, your delta mu has to be like on the order of like 15 kT in order for you to start seeing some bang for your buck, like getting, so that if you now drive it more and more, you get higher and higher efficiency. Like up to here, I mean, most people would like, if you were to ask, what does near equilibrium mean? You might say near equilibrium is like when delta mu is on the order of kT, right? Where, but that's like nothing, right? Like here you have to get like 10, 15 kT for you to start seeing this, the inflection point. And so the solid symbols are just what, like if we, from the known values of delta mu in these organisms and the measured values of efficiency, where we just place these different molecular machines or chemical engines, right, in vivo. But this is the only in vitro data that we have. And this is not a fit, this is just superimposed. But what's interesting to know is that this explains qualitatively, I think, a very interesting feature that you definitely see in the experimental data, which is if you make your ATP concentration not this high, which is very, very far from equilibrium already, you essentially get like non-operational, like your dining essentially doesn't really walk. Okay, so its efficiency is super low. And so you get nothing over these spans of delta mu. So if you think about this evolutionarily, right, like if you have something and you evolve some primitive cell and you're trying to, like, you know, convert energy, and you're getting energy however you can, and you're not getting that much energy, and you're trying to convert it to some delta mu, and let's say you're using some chemical source, some primitive version of ATP, and you work and you work and you work, but you just get your ATP concentration such that your delta mu is like 3 or 4 kT. You might still be able to do very simple things, but you have not activated the suite of molecular machinery until you get, so like basically the cell has to wait an evolutionary time until it has acquired a sufficient chemical potential driving force such that these classes of chemical engines can be efficiently activated, right? So I think this you know, presents like a way to think about like a sufficiency condition from, you know, that a living system must have. It must have the delta mu above this threshold that's set by the essentially, most importantly, by the load resistance, the kind of load that you're trying to achieve. Okay, there's so there's this kind of a sufficiency divide between like living and non-living systems, and it's also, I think, interesting thing about in terms of an evolutionary constraint, until what had to happen, like there's no gradient here, like so like if you're here, there's nothing you can do to make it better, right? You just have to like evolve for whatever reason so that you're here, so you now actually have a gradient to like evolve a higher and higher delta mu. Here, the delta mu is so small that you don't even have an evolutionary gradient to evolve a higher delta mu. So there is this kind of threshold that you have to surmount. Now you might look at this plot and also say, okay, well, what if I just have RS over RL to be really small, right? So that I'm driving really large loads, right? Because then it looks like I can just get really high efficiency even when I'm close to equilibrium, right? So then again, that brings back to the analogy or the issue that we had with the kinetic proofreading. There's must be something else you're giving up when you're here. You can't be close to equilibrium and seem to have like pretty high efficiency. And what are you giving up? You can actually convert this equation into this one, which is a trade-off between energy efficiency and time efficiency. So what is F? F is the net forward flux and B is the net backward flux. So F minus B is the total forward flux, B is the total backward flux. So F minus B is the net flux. That's the current that I was talking about. That divided by the forward flux is what we call the processivity. Okay, so if that's small, that means most of your forward flux is getting like undone by the backwards flux because all of these processes are reversible, right? So this is a measure of your time efficiency. And if you now apply your time efficiency versus your energy efficiency, you see this trade-off, right? So now it's only when you're very far from equilibrium that you actually can get rid of the trade-off. If you're so far from equilibrium, you can have time efficient and energy efficient things, but you have to pay a price that you have to be very far from equilibrium. When you're not far from equilibrium, the cases where you're seeing the high energy efficiency from the last slide, you're paying for that by having low time efficiency. Okay, that's not what's shown on the last slide. So that's the price that you pay. Okay, so I think that sums up kind of the things we're trying to go. So here in this example, I think it's clear what the effective variables are. These are the circuit mapping of the resistors and battery, right? And I think we can now predict analytically things that are not obvious without understanding these effective variables. And that's not only that far and near equilibrium are different, but it also tells you what kind of different properties get activated, and also tells you that surprisingly, both in the specific example, like the in the microtubule case where you go from logarithmic control to linear control of the catalyst, and more generally, like in the efficiency transition inflection point, when you're close to far from equilibrium, that it can be a relatively sharp type of transition. That's you can suddenly be in a regime where a living system looks different from a non-living, like it could have a proto-living ancestor that's not very different from what it is. It's just it just evolved to have like a higher delta mu, and suddenly it looks a lot more lifelike, right? So, but we've explored and are in the process of now using this to build something, because I think the easiest analogy to see is like, okay, if we have something into a circuit formalism, you should be able to now, in principle, leverage, what people have been able to do with electronic circuits, right? Like the iPhone, that level of scalability of building something with modularity and robust design that you can go to have something that have so many parts, right? I don't think we can get to that level of like precision and control modularity for biology, but can we get further using this kind of effective collective variable? That's what we are trying to build. And hopefully we'll have something to say about that in the future. But I hope to motivate that in principle, it is possible to build these things if you can first do these first two steps with my kind of mini example on the neural circuits that I kind of led this talk off with. So I think with that, I'm at exactly 1 hour. So we have like half an hour to kind of discuss stuff.
Yeah, absolutely. Thank you so much, Milo. That was absolutely fascinating. I want to start off with a scientific question. So in the last segment of your talk, could you say something about whether anyone has tried or whether it's possible to really measure this quantity RL over RS?
Yes. Yeah. So let me see if I can. Why can't I go back? Let me just escape this. Hold on. Okay, so yeah, Oops, there we go. I can do this. Okay. Yes. So the yes, the RS over. Yeah. So as I mentioned before, it is in principle possible to measure these because like for this system, for example, right? Like if you have some system now and you have an in vitro construction of the system, and the system, let's say, runs on ATP, you can have the system be at equilibrium and dope in a small amount of ATP and measure the perturbative response, the linear response of the current to your the ATP. That linear response will give you the RL plus RS. Okay. Now you should, now the RL is the one that's generally very complicated because that's some molecular machine doing stuff. Your RS should be, the elementary parameters of a RS should be individually like ascertainable, right? Because the RS has to do with, like, for a molecular complex, right? What is the free energy difference of bound and unbound of this bound unbound to ATP, and what is the characteristic like rate constants, right? For this kind of event. So because this is more universal, because lots of things share the same battery, you can kind of do a lot of work to make sure that this thing is individually like well parameterized, and then you can just measure RL plus RS through this perturbative measurements, and now you have both RL and RS. So I think what makes this actually amenable is that this theory applies far from, so the parameter, the RS and the RL, is the same RS and the same RL far from equilibrium, but you can obtain what their values are at near equilibrium.
Right.
Hi, yeah, I just had a question trying to build intuition for the kinetic proofreading. Could you say,
Oh yeah.
from an intuitive or from a series of molecular events, what the waste would correspond to? Is that kinetic? Is that enzymatic steps, or is that just binding, unbinding?
Yeah. So the waste has to do with, so there's 4 currents here. Can you guys see this? So there's a current. So the best way to visualize this is the way that we visualize this in circuits is in terms of like loop currents, right? So you have one. Let's consider the steady state. You have one loop current that's going clockwise here, and another loop current that's going counterclockwise here, right? And there's another loop current here and another loop current. There's 4 loop currents, right? So the error has to do with the loop current, this loop current versus this loop current, because this loop current is the production of this net, like bad thing, right? Where you put on the wrong amino acid. So that's the error part. And then the waste has to do with basically incorporating the fact that there's these processes, right? So these processes are part of the proofreading. That's what makes the error low, but you pay a price that you can be wasteful. So sometimes you will have a thing fail even though it's correct, right? So the waste comes in both if you go in this direction and that. So if you bind the wrong thing, right, there's still a chance that you'll hydrolyze that GTP, and then it'll fall off. So nothing has happened, but you've wasted a molecule of GTP. The waste also so that's this loop current. You can also waste it here where you actually have the right thing and everything's fine. You've hydrolyzed the GTP, but just stochastically, instead of like doing the right thing at the next step, the thing happens just fall off stochastically. And now you've done nothing again, and that's also a waste.
Okay, I think this makes sense. So the argument is that about the interactions in the ribosome could have evolved to increase that waste term, even if it does find the right thing.
Yeah. And in fact, the best it can do is this. So like the best it can do is basically this, that like whatever you gain from error, you have to compensate for by waste. And you can, your system can decide to portion it in different ways. And in fact, I didn't mention these data points that are experimental data points are under different conditions. Okay, I think in this case, it's different condition of like magnesium or something. So under conditions of maybe signaling relevant conditions, right? Your system can choose, and it does choose, different values of the trade-off. It can say that in this context, it's okay to be wasteful because we're rich. And in that context, it's okay to make some errors. And it does, right? So the best it can do is that, and this data just shows that the system has been optimized to be at this best level of trade-off. Because if it's not optimized, it will actually be below this, where like the parameters are not optimized to like get as good as this.
Interesting. Great. Thank you.
Yeah, this was really interesting. Thank you so much. I was wondering, I'm kind of curious, like how you landed on this framework of circuits. Like, it seems to me that maybe the standard paradigm is like drawing like a graph of nodes and edges or something, and maybe writing down some probability weights between them. And this adds, at least it seems to me that it can incorporate passive and active elements that maybe you can't necessarily have in that graph structure. I'm sure there are other aspects of it. Can you say a little bit about that?
Yeah, that's a good question, and I think that's also a good point to speak about what is this construct useful for, right? So just like there's no free lunch overall. I mean, even in my talk, I show that there's a price to pay. There's also, this framework is not optimal for everything, right? So we believe that. So to answer your first question, I think the one of the key differences that we did here doing this is to really think completely in terms of the reference equilibrium system first, and do everything relative to the reference system. Okay, but related to that is like, that is only like an intuitive way to go when the driven transitions are relatively sparse. Because if you have batteries everywhere, at that point, like your reference system is like, right? And in fact, that's borne out by this. So this circuit theory is true. Let me go back to my initial slide. Sorry, I have to do my sliding thing, so it's going a little slower, but just so that we're on the same page with the equation. Okay, yeah. So this mapping is all good. This equation is true no matter how complicated your state space is. But it is only useful when the state space is sparsely driven by batteries, right? Like when there's batteries everywhere, then it becomes less useful, because then there's less like blocks of resistors to coarse grain, and you also lose some analytic tractability, right? There's some theorems that you can still apply and some perspectives that are still good. For example, you can still treat any two-port like some kind of input and some kind of general output with a bunch of messy stuff that can have lots and lots of driven transitions. You can always map that to a Thevenin equivalent resistor that is in series with a Thevenin equivalent battery for any complicated. So that there's still useful things you can do, but I would say that a lot of the power of this representation is lost when you're given things that are very densely driven. But I think that that's something that is also evolutionarily maybe relevant, right? Like sometimes when you find like constraints or design principles, if these are really good constraints or design principles, we might also expect that the evolution itself has been bound by them and maybe has evolved according to that, right? So the fact that a lot of these molecular systems have been are driven by these in many cases just a single energy source, right? And then do a lot of things. Maybe what allow them to modularize and to evolve very efficiently. Because if you think about it now, if you just have a few, like the intrinsic qualitative behavior of your circuit is now dictated by the topology of where the energy comes in. Everything else now can be coarse grained. And so now you can have a system that seems like has lots and lots of parts, but if you mutating or evolving things in a big passive area, all you're doing is just pushing some effective parameters around. You're changing the effective parameter, but you're not changing the qualitative construct of the circuit. It still has the same circuit topology. It's not going to have any surprising qualitative like switches or whatever, right, behaviors. And in fact, if you even quantitatively, by making mutations inside like a dense part of state space that's not directly driven, their effects might be highly diluted and compensated by many possible redundant effects that compensate. Because at the end of the day, if all that matters is the value of a large Thevenin equivalent resistor, you can change all of the resistors in that equivalent resistor in different ways to get the same change in the total resistor, right? And by the same token, you can, if something happens to change the overall resistor by some way, there's many paths to correct that in changing many of the other resistors there, right? So, and I think, like we haven't done a formal analysis of this kind of thing, but I think this kind of idea or framework is also consistent with, like what people like Jamie Sifna has been talking about in terms of sloppy and non-sloppy modes, right? Like there's certain things that are highly redundant and other things that are more sensitive, right? You can imagine things that are like now topologically placed relative to where the energy sources are, that could be a much more sensitive thing and have fewer ways to correct for, right? So I think that's that's something that we think is a constraint of this formalism. But if this is really a good effective variable, this might also be a way that it has also constrained the evolution of these systems themselves.
Yeah, no, that's very cool. It's like a robustness control. Yeah, very interesting. Thank you.
Okay, so why don't we just switch modes a little bit? I'm going to